Today, we have a wonderful panel who will be speaking on innovations across the cell therapy workflow from apheresis to analysis. My name is Linda Barnes. I am a consultant and a member of AABB. I work in the biotherapy space, and I'm so excited to get us together to talk about our innovations and from apheresis to analysis. So I'm going to go ahead and sit down now. And maybe we could have our panelists introduce each other or introduce themselves. Why don't we go ahead and start to my left here? Felix Montero, do you want to talk a little bit about yourself and how you came to be here today?
Sure, absolutely. So, good morning, everybody. My name is Felix Montero Julian, and I am Vice President of Scientific Affairs for a company named Apheresis. So, I have a PhD in immunology, and I have been working in the pharma industry for more than 35 years. Working 20 years for Beckman Coulter in the flow cytometry field. Then I moved to bioMérieux for at least 13 years, working in the development of analytical methods for cell therapy products, in particular for sterility testing, mycoplasma, endotoxins, and so on. And since 4 or 5 months, I moved from France to San Jose to San Diego to work with the Axelix. And I will tell you a little bit more about Axelix, a new instruments for immunophenotyping of leukoplaques used for cell therapy for us. And I am very happy to be here. Thank you.
Thank you very much, Felix. Why don't we move to you, Jenny? Tell us a little bit about yourself and how you came to be here today.
Sure, thank you. I'm Jenny Sander. I am a solutions manager at WellSky for their biotherapy solutions. By way of background, I worked at the St. Louis Cord Blood Bank for 8 years in St. Louis, Missouri, and then I transitioned over to WellSky and have been there for 13 years. I currently work with our engineering team and our services team on helping build solutions for the cell therapy industry. So we have a full suite from collections all the way through processing and infusion and reporting that we work on and work with our clients to build the solutions in the way that you guys need to enhance your day-to-day activities.
Thank you so much. And it's wonderful to see a cord blood banker here now that cord blood banking is becoming part of the AABB annual meeting. We'll move to you, Trevor. Welcome to the Biotherapies Pavilion, and maybe we could hear a little bit about you and what brought you here today.
Sure, thank you. Happy to be here. I'm Trevor Smith. I'm a global marketer at Terumo Blood and Cell Technologies. So a few of you in the audience might have heard of a Spectra Optia, that we provide that, obviously as a device solution. By way of background, I'm actually an immunologist by training with the scientific and crossed over to get an MBA and get more into the business side of life sciences. I've worked at GE Healthcare, now Cytiva. I've worked at BioIVT, and happy to be a part of this panel today to talk about cell collections.
Wonderful. So it sounds like we have both the analytical component, we've got the information technology component, and we also have apheresis devices that are involved in collecting the cellular starting material. Maybe you could tell us a little bit about your experience with some of the challenges that you see in the continuum of the biotherapy's life cycle from cellular starting material collected by apheresis through to the identification of what the material is, and then how the information and technology gets shared amongst the various stakeholders. So why don't we start with you, Trevor, and talk a little bit about apheresis and some of the interesting aspects of the devices and the connectivity of those devices.
Sure, happy to. So with apheresis, I think each product is very unique because each patient, each donor is going to be very unique, right? And so the device and the hardware that we create is designed to take away some of the variability that you have in those starting materials just inherently. And so when our device performs well, you get the best possible collection that you can have. But that's really, you see what comes off the device in a bag of cells, but actually the devices itself, capturing a lot of data that can be critical to the analysis of the process of collection. And when you marry that data, not only to like, okay, what happened during the procedure with the device, but also to who was the person hooked up to the device, what did the cells look like in the bag coming off the device, and marry all these data sets together in a comprehensive way, you really start to get, insights into how to do collections even better and make them more predictable. So it's very exciting this in the space. So
Wow, you talked a lot about data from the collection process, but I know Felix, you had talked about data from the donor and qualifying the donor. Maybe you could share a little bit about what you see with respect to the analytical process.
Sure, yeah. the, so far, the leukapheresis products are mainly characterized using the CB6 one. So, and there is a more request about having a more deep characterization of these leukophage products, in particular for the immunophenotyping. These requests come from the developers mainly, in which they really wants to know what they have in the device, how many T cells, how many B cells, how many NK cells, what is the CD4 CD8 ratio they have, right? In order to take some decision and start to make these products as the beginning. So all this data has been generated so far, but sometimes this data has been delivered too late to really help and take some actionable decisions. And this is what we are trying to do in our company, is really to have and to master the flow cytometry technology that is a really cornerstone technology for immunophenotyping of different cell populations in blood, right? Beyond the TBNA cases, also the CD34 cells, for example. During the leukapheresis mobilization, that is also critical to know exactly how many CD34 cells we have in the bags. And at some point during the collection, take decisions, we continue collecting or we stop that, right? So today the technology is there, and but this is not really fitting for purpose. And this is what we are trying to do today in our company, is how we can provide tools and analytics that can help the collectors and the developers to better characterize these leukopacks or these leukapheresis products that can bring value to them at the end of the process.
That's very interesting. So you're giving more real-time information.
Exactly. So, and I think that the the future of the technology is obviously smarter, but also we need to get the data at the point of collections to really take better decisions in the way we need to manage these different problems.
Interesting. And Jenny, I know that a lot of this comes down to the transfer of information between various stakeholders. So even if we get the information collected from the apheresis device and from the analytical process, tell us a little bit about how it all comes together so everyone can see it.
Right, thank you. So the biggest thing with integration that is key. That is what we get asked. Number one question from every client that we work with is, who do you integrate with, and how quickly can we get that set up? We are working with Terumo on an interface with the Optia, between our solution and the Optia, to be able to pull that information directly from the machine. So that way, with that interface, those collection details are automatically coming through in your solution, and you're able to see that information in real time. You know, we know timing is precious with these cells, and having that information come through immediately is critical. Additionally, not having to re-enter anything by hand. You want everything from a quality perspective to come directly from those machines and those instruments. And we focus on that to be able to pull that data as crystal clear as we can.
And how about the transfer of information from laboratory analytical devices? How do you manage that as well?
Yeah, same process. So we have instruments hooked up to our processing procedure selection, so that way, if you have, for example, a Sysmex tool, instrument or something, those critical cell counts are coming directly from that instrument and populating the software in real time. So you're able to pull those values down immediately and then run those automated calculations based on those values.
Interesting. So what are some of the challenges that you see when you're working with your clients that they express to you? And maybe I'll start with you, Felix, and then we'll go over to our other sofa. So Felix, what are some of the challenges that some of your clients share with you about about the process problem you're solving?
Yeah, historically, flow cytometry has been a very interesting technology, but very sometimes cumbersome to set, right? And you need to have a very high skill sets people to run the flow cytometers and to do the analytics. So with the scarcity of all these different resources, so what the customer is asking is really to have a simple to use systems that you don't need to have is a PhD to run a flow cytometer, right? And I'm really having able to do any particular person working in the lab that can run the assay, and the instruments can give you the automatic results after the analysis, because this is what we are trying to do, to avoid to do all these manual analysis that sometimes happens when you run flow cytometry data. So developing some specific auto-classification algorithms that can analyze automatically the samples, I have the results after 30 minutes, right? And for me, I think that the integration of the Apheresis parameters, the CBC plus the immunophenotype in this can create a huge value in this donor characterization. And it's not only for the autologous approaches, but also for allogenics in particular, right? So some customers or some developers that are developing, for example, NK cells, and they want to have leukocytes containing a lot of NK cells. So using databases that can be populated with all this data will be extremely beneficial and very powerful for the developing of these therapies and for the management of these different cases.
Interesting. And Trevor, what are your thoughts related to the integration that Felix was talking about how could that potentially optimize collection of the PBMCs or the MNCs?
It's a great question. I mean, the device is powerful that it captures its own data, but really the cells coming off of it are a true blind spot to it, right? Like, what do the cells look like? Like, how are they not just at the end of the collection, but also at a midpoint of the collection? And with something like the Acellix device, right, you can begin an apheresis procedure and actually take a sample and see in real time, like, are we hitting the target that we're expecting? And it gives you an opportunity to make adjustments, right? And because especially in a clinical setting, you have one shot on goal many times with doing this collection. You want to get it right the first time. And so every opportunity that you have to kind of recalibrate and make sure that it is a success at the end, it's huge. So, we've talked with Acellix quite a lot about the applications for it, and it's just kind of a natural harmony to it to just have the visibility to what it is that you're doing. So
That's wonderful. I'm glad to see the collaboration that exists here amongst our panelists. I'm wondering, what do you think, Jenny, with respect to also visibility within the institution and potentially to the sponsor or the downstream manufacturer who's going to be using these cells? What are your thoughts about how that information could or should be shared with in order to do either process improvements or aid the manufacturing process?
Yeah, I think tracking and trending of that data is key in moving forward with those sponsors. Being able to run your own reports, pull all the data points that are again coming from those instruments into the solution, and build that out and send that out automatically to those sponsors is something critical that makes everything easier on everyone invested in the product.
Yes, we've often heard about, the universal portal, and I'm thinking of my experience working in apheresis unit and how challenging it was to think, okay, this collection is for which sponsor, and then which portal do I need to use, and what parameters are they requiring, et cetera. Tell us a little bit about how WellSky's solution might speak into that, or what do you think about it as a programmer and an expert in this field from the technology perspective? What do you think about when you hear, oh, well, we want a universal one that solves all of our problems?
Well, everybody wants universal everything to solve all their problems. That's number one request. But basically, being able to track those studies in our application, which you can, tracking the studies and the parameters that need to be included, the labeling associated with it for each sponsor, being able to print off that data or send it, that is able to be done within our solution, and then be able to send that out accordingly.
Wonderful. I just want to let the audience know that we're going to be opening this up to questions from the audience in just a moment. So maybe cue your question up and we'll go ahead and pass the mic around. So be ready. Let's see, Felix, do you have some additional things that you would like to share, given your vast experience in this field, about what we should be looking for in characterizing the donor material? Because we have a variety of different things. You mentioned NK cells, CIK cells, the list goes on, G minus NK cells, it goes on and on. So tell us a little bit more about what you see.
I think that one of the main questions we have today is, so we have different collector sites, right? On every single site could be different both, have different procedures, different ways to collect, but we need to have a common analytical procedure to be able to compare data, right? And standardization, I think that this is a key word now in the field. So how we can standardize processes, SOPs at some point, so the collectors can have access to different samples, having the same characteristics, or at least having the same kind of critical quality attributes measure to be able to take some decisions. So beyond the characterization of the cells, I think that the main part is the standardization. And this is also something that we are working on. So trying to characterize our instruments in order to have different instruments providing exactly the same results in function of the different sites they are using. Characterization and standardization is key for me in this decentralized model too, right? Because many developers have access to different kind of products from different suppliers, different collectors. But sometimes the products are not the same or are not characterized the same. So the goal is really to be able to have and to the process in place to have the same products coming from different sites.
Well, I think you're speaking with the right audience in terms of a standard setting and education organization. It really makes a lot of sense what you describe. And when you think about a company that's going to market and trying to commercialize a material or make sure that they get the equivalent results for patient A and patient B, it's really important to know that you've used a standardized method. Appreciate that. This right now, if you just joined us, we're having an open mic session where we're talking about innovations in cellular therapy from apheresis to analytics. We have three excellent panelists here who are talking about their experience, analytics, information technology, and the apheresis devices themselves. So I'm going to open questions up to the audience now. When you think about your experience in the apheresis to analytics experience, meaning the all the way through to the point of manufacture, what are some of the questions that you might have of the panelists here? Or if you have some experience that you would like to share, feel free to do so. Let's see. Go ahead and raise your hand. Just hold your hand up if you'd like to take a mic and either make a comment or ask a question of the panel. Let's see. Don't be shy. It's a very comfortable environment here. But maybe while you're thinking, why don't we go back to talk a little bit about the process of designing a device that collects apheresis starting material, many ranges of cells. What are some of the considerations that you think about when, from a Terumo perspective, when you're developing the apheresis Spectra Optia, for example?
Yep. Well, I know Spectra Optia has been around for much longer than I've been at the company, so I know the design was certainly elegant. I've seen the product at the end of it. But one of the key themes for really any kind of device that we developed for the handling of cells, the collection of cells, the processing of cells, it's really the reliability of it. And we want to bring a particular kind of innovation to it. We want to advance the space. We want to make sure that the cells get to where they're supposed to be in the way that they're supposed to be throughout the whole life cycle of the process. So if you think about a canonical kind of cell therapy workflow, oftentimes it begins with Spectra Optia and actually collecting the cells, but then we also have downstream manufacturing devices like the Quantum Flex cell expansion system for growing the cells up into the therapy that's going to be, or the Finia fill and finish that it's the final step, readying the end product for infusion into the patient. So it's really nice to see the whole process flow through in a single portfolio. That's like, this is the journey that the cells take, from in an autologous space, like from the patient back to the patient, and it's our devices all the way through, and of course welders as well. I think a lot of people are familiar with our welders and actually keeping those connections sterile and reliable too.
That's really helpful. How is that the same or different, Jenny, from your experience in the product development life cycle? How do you get feedback and incorporate what the people in our audience here might experience working with your technology to improve it?
Yeah, for sure. I mean, as we know, this industry seems to change every day. So there are new features and requests that come through consistently, and it's our job to work with everyone to try and make it as universal, like we said earlier, as we can, and make it work for everybody, but also be extremely configurable. Because again, we know everybody does something slightly different. And by giving you guys the ability to configure things yourselves and build those forms and sets yourself up for success. So we love to work with you guys in real time and get that feedback and then incorporate it into our releases.
And you have a relatively new device. How do you anticipate getting feedback from clients who are using it?
So yeah, we have a process to discuss and collaborate with the customer in the implementation of the system, in the validation of the process here, and we are learning all the time also, about the needs they have. So some developers express the needs to have the real value before cryopreservation, for example. Some really wants to have values at the moment they receive the local packs in their facilities. Some others wants to have a full validation packages for the implementation of the technology, etc. So we work in different areas with them to capture and master and take all the benefits of the platform as given to the users.
That's really, really helpful. Maybe someone in the audience could share a question. Thank you very much. You got it? Thank you. Go ahead and say your name and where you're from.
Hi, I'm Kimberly Banks from Gulf Coast Blood in Houston, Texas. Quick question. I think it's great that you guys are working together for that interface from the Optia. We know that Optia is used across the industry. And my question is, are you the need to build more interface on the Denver or the interface be different systems rather than this?
Yeah, I think it's a great question. Like the interface with Opti and the Acellix device, for example, like that can just be off the shelf, right? As long as you have both devices at a single site. I think the kind of interfacing and connectivity, and Jenny with WellSky, is having this common software platform that can be something that the data goes up to, that can be drawn down not just from a single site but multiple sites in a network, and those insights can be used like across a single organization. Like that is being done through partnerships for sure. We're also working on connectivity of the device, making sure that the data can push and even pull back. It sure is. So yes, we're trying to advance the device as well to do that, but it plays very well into ecosystems. Yep.
And then just one other question for the ACELIC. Is it fairly a lookup instrument for test the lookup pack? And then are you looking to expand that capacity to other products?
Yeah, so the platform today, obviously, we are talking about local packs because we are in here. It's home for us, right? About flood bags, polylifer packs. But so the beauty of the technology is that can we use all the continuum to the value chain, let's say, from the local pack characterization, cryopreservation, characterization during the development, and then the technology can be used also to do the in-process testing during the manufacturing process. For example, we have customers using it, developing some CAR T cells, and they use the technology for the process testing during the transduction steps. You know how the virus has been be effective to transduce the cells. And we have also others that use the same technology to do the product release at the end, right? So the technology can be used all the value chain of the cell therapy products, from cellular characterizations to product release, end to end. And what we like also is the fact that using the same kind of technology, you can compare data, so you can compare all the data in the process and have it exactly the same technology doing and getting all these different results. So Apheresis is one, but we also work with immobilized Apheresis. We work also with PBMCs, cell lines, etc. So different kind of products.
Very great question. Yes, we have another question. Excellent. Thank you.
Hey, Chris Law from Life South Community Blood Centers. There's a lot of discussion about characterization of the product after collection. Is there anything going on in characterization of the donor prior to collection in how good, how high quality their product would be?
Great question, Chris. Thank you.
Yeah, there are some works ongoing on that, So to have a more deep characterization of these different donors, in order to characterize it, right? So to see, in particular, in the allogenics part, not in autologous is not the case, But in allogenics, absolutely, yeah, there is some works ongoing to try to identify what could be a good donor, right? So in particular for cell therapy.
I can also add more to that. The pre-collection characterization as well. We've even done work in optimizing collections and being able to look at, okay, you got the pre-collection counts and what came off the Opti at the end of that. You can actually format the data to kind of inform on how much like whole blood volume the Optia has to process to hit a certain number. So it's really interesting. It's just, the more data, it's like gold, and then to be able to kind of get that algorithm going. Great question. Yeah.
And I'm interested in how that information could potentially be retained as part of the collection record. I don't know if you have some thoughts on that, Jenny, how that might be contained, particularly if a donor might have serial collections, how you could compare one collection over the other.
Yeah, for sure. So within the donor piece, you have different collection events, and you can configure those forms to pull all the data from those machines and then compare those events over time. So they have specific records for each collection and then compare those again through reports.
Great question. Thank you, Chris. Any other? Let's see if there's another question in the audience here. We have a question over here. Thank you. Feel free to identify yourself and where you're from.
Thank you. I'm Sarah Lewandowski from Terumo Blood and Cell Technologies, and I think I have a question mostly for Jenny. I'm curious if you're fielding a lot of concerns about data security. So all that data that's stored and transferred, that's something that we're sort of figuring out how to handle. So I'm curious about your perspective.
No, we haven't really had many questions as of yet related to that. I mean, we do have a lot of, like I said, existing interfaces pulling that data off those instruments and machines. And obviously, we keep that in our environments at the sites. So that is protected with security protocol and everything else. So as of yet, we've not, but it's not to say we won't.
Well, we certainly hear a lot about cybersecurity. It's an excellent question. And I know that is something that's part of AABB standards as well, is making sure that you have a robust system in place and that it's tested. And I see Kate here nodding, who is has led our cellular therapy services standards. Kate, do you want to make any comments on that? I covered it, but you can say it far better than me here.
Kate Brown, I'm the chair of the Cell Therapy Standards Committee. So as we were discussing, I mean, I think making sure that your facility has in place a system to test all your data security and understand what that looks like and making sure that it's sufficient is going to be really important as we see sort of an increased amount of data being collected and stored. So yeah, that's something that your facility should definitely be looking at.
Thanks so much. Didn't mean to call you out, but you've done a wonderful job. Thank you so much for leading the cell therapy standards. Appreciate it. We have other questions from the audience. Thank you. Please just identify yourself, where you're from.
I'm Maria Roots, Gulf Coast Blood. The flow cytometry is a gold standard. So how does Apheresis correlate with the flow?
I mean, yeah, absolutely. Great question. And every time we receive the same question, right? So we have a lot of data showing that the Axelix instruments correlates perfectly well with the regular flow cytometers, right? So the instruments are equivalent. We have a lot of data with different kind of samples showing that the two systems are equivalent. We compare with MaxQuant, FaxLyrics, FaxCanto, etc., and the data are equivalent. No issues on that.
Thank you very much for that great question. Again, pulling it back to standards, the requirements for instrument IQ, OQ, PQ is really important. You've highlighted the value of doing that. Are there other questions that people have when they think about from apheresis to analytics and analysis? What are some recommendations that you might have of things that we should be really actively pursuing in the future so that we can make our lives easier and less of a swivel chair where we're keying in something here and then moving over here and adding it in a paper-based system that I know I grew up on in the apheresis unit, and even batch records are still primarily paper-based. What are some recommendations that you have that you'd like to share? Some pain points, perhaps. Aw, nobody's feeling any pain out there.
We're doing our jobs.
Yeah, there you go. Good. Well, what do you hear from your customers, Trevor, of things that could be done better and differently?
Well, I think, as highlighted before, every patient, every donor that's hooked up to a spectropty is going to be different. So variability is going to be the enemy. So one of the solutions that we have is actually working with them and getting our arms around that data and doing that analysis for optimization. It's a service that we offer. It's something that we've partnered with many people and continue to partner with. Not just in a stem cell transplant space, but also CAR T and those collections. It's really something to see. I referenced it earlier about how you can, do this analysis and marry pre-collection counts to whole blood processed on the device and get this like really neat line of what you have to do and informing on your process. And it's just the limit is only how you want to slice the data. So
That's great to offer that optimization process you were just talking about. I see in our audience here that Jeff Wren has joined us. There's Jeff, who is a vice president of biotherapies for AABB. Jeff, is there anything that you'd like to add to the conversation here related to the need for standardization, harmonization, optimization of the process from end to end?
Putting me on the spot, Linda, I would say that one of the pathways for us to be able to scale out some of these really impressive therapies to increase access is going to be through technological innovations, right? So we've got the science down, that works great, but how can we scale that up? Working with partners like you guys, getting you guys out in the community, getting feedback from a clinical side from the developers, rooms who are going to help us do that. So I really appreciate you guys taking the time this morning to share where you're at, make sure these people are aware of what's going on. I mean, people read the headlines when the science comes out, when the new therapies are showing that they work. Maybe not so much, on the equipment, on the supporting supplier side, but they should, because when they realize what you guys can provide, light bulb goes off. All right, well, now we're able to give these to the patient. So thank you for that. Appreciate this time, guys.
Absolutely. I want to give our panelists one more chance to make some closing reflections on your experience in the field and what you see in the future.
So from my side, I'm very excited, so seeing the field evolving in the way we are moving. And for me, the most important thing is seeing patients get cured. This is what motivates me every day, So analytics are key. I mean, we are blind if we don't have analytics that measure exactly what we need to measure, right? And if we don't measure, we don't know what to measure. So as an analytical person, this is critical. Patients are behind in every decisions we take. And for me, the destiny of every cell therapy product depends on the characterization of the cellular materials. So we need to characterize these products better. And we need to talk to the developers too. It's key, because we need to know exactly what the developers want and how the collectors can adapt also to reach the level of criteria that are needed to have robust products, potent products, and safety products.
Thank you. Well said, Felix. How about you, Jenny?
I'm going to say a buzzword, but I'm going to say it anyway. So I'm going to say AI. I think it is critical, especially in our field, to use that to the best of our ability to make these solutions better for you, to enhance them quicker for you. Make it so that, if you're trying to build a calculation for a cell count, you don't have to think twice. You can ask the solution to say, hey, I need to figure out this calculation. It spits it out. You put it in the solution and then you're good to go. Again, timing is key. You don't want to waste time on little things like that. So really using artificial intelligence as much as we can, integrate it into our future, it will be beneficial to all.
Interesting. The AI comes up. All right, over to you, Trevor. Trevor, go ahead and close us out.
Yep, will do. So when I started in cell therapy about 10 years ago, it was strictly on the manufacturing side. And the view then was for the manufacturing process, you get what you get and you don't get upset. And it's the responsibility of the manufacturing process to fix any deficiencies in the starting material or any variability so that you get a reliable therapy at the end of it. Um, since coming over to Terumo and getting more on the cell collection side with BioIVT, you start to realize that there are so many levers that you can pull, like as a therapy developer, that you should consider, starting materials as an extension of the manufacturing process, if not outright the manufacturing process. And do you have to engineer such a significant manufacturing process to fix deficiencies if at the start of collections you addressed a lot of them to begin with? And so I think this conversation is happening in real time. I go to cell and gene therapy manufacturing trade shows. They're starting to talk about collections. I come to collections trade shows. They're starting to talk about manufacturing. And it's really just nice to see these paths finally converging, the hams talking to each other. And I think it's only going to be better for patients moving forward.
Excellent. Well, that's it gives me a lot of pride to think about what you're doing to enable that and what all of the people in the audience are doing in order to support patient care, leverage new technologies, and then I love that, the two hands talk to each other. So thank you, everyone. We'll be milling around up here in the front if you want to come on up and ask questions. But otherwise, have a great meeting. Enjoy yourselves, enjoy your colleagues, and thank you so much for impacting patients' lives.
Thank you.
Thank you.